the title, just this situation for when people are coming in.
Mm-hmm.
Okay. Oh. Let me know when I should start.
You're probably good to start whenever you're ready.
Okay. Thank you all for coming to this session. And I'm going to stop sharing. You are in the faculty seminar, Regenerative Engineering. I'm the moderator, Elizabeth Wayne, assistant professor at Carnegie Mellon University. And I just want to thank you again for coming. Each talk is allotted 15 minutes, and I will prompt the speaker when there's 1 minute left. There will be an additional brief 3 to 4 minutes for Q&A. And if you have additional questions, I'm sure the speakers will connect with you via the HOVA chat and provide their contact information for further correspondence. This session is also being recorded by Zoom. Our first speaker in our regenerative session is Dr. Justin Brown. And he will be giving a talk entitled In vitro quantitative evaluation of MACs from 3 unique donors in response to macro, micro, and macro, micro, nano rough titanium. I'm sorry, he's going to do a way better job of talking about this than I will. Let's all thank and welcome Justin. Thank you.
Elizabeth, thanks for the intro there. Let's see if we can hop over and get to sharing. I'll say that the macro, micro, nano moniker is certainly a little bit of a tongue twister, and that was not necessarily my doing. That was at the urging of the sponsor for the research, right? So you sort of lean to what they the phrasing they would like to use at times. Anyways, my name is Justin Brown. I'm an associate professor kind of just a little bit east of you guys in Pittsburgh at Penn State University. We had hoped to host NEBEC about this time last year, and things happen. I'm sure you guys are all aware, but here we are today, excited that we've got it back and rolling. These virtual conferences have been a lot of fun. A lot of interesting moments. The exciting thing for me is I get to finish this talk and then teach a class. But that's okay. Anyways, so yeah, the title is In Vitro Quantitative Evaluation of MSCs, or Mesenchymal Stem Cells, from 3 unique donors in response to macro, micro, and macro, nano rough titanium. This was some work that we did pre-COVID with a couple of undergrads in my lab. Anastasia and Karina were two undergrads within the Honors College here at Penn State. They've both gone off to medical school, but they were a great help, turned into a nice little data set. Got to figure out what button to click to advance the slide. There we go. All right. So briefly, I wanted to give you guys some background on kind of where we were coming from with this project by setting it up based on what we've done before. And our previous efforts looked at both the early and long-term response of stem cells to these rough titanium surfaces to see A, if cells were able to attach and spread and migrate in a timely manner, and B, if they start to differentiate. So as engineers in the regenerative engineering space, we often think about the key feature of success for a scaffold or a graft being whether or not the cells differentiate, make our target tissue, but a surgeon often has a dramatically different perspective. So what's of particular importance to them in the ortho space is, will that graft encourage the migration and proliferation of somatic or stem cells prior to the bacteria that's inevitably introduced during the surgical process? So they call this sort of the race for the surface. So a lot of our early efforts on these titanium surfaces were looking at those early events, both attachment and migration. And so here we've got a couple of videos playing. The videos, I'll sort of detail real quick and then tell you the rest of the story. The one on the far left is a mesenchymal stem cell on peak, polyether ether ketone. The one just to the right of that is on smooth titanium. One more over to the right is on a macro micro titanium. And the one on the far right is the macro micro nano titanium. So if you watch these cells migrate, this is over the same time frame. You see the one on the peak doesn't tend to move very much. And this is starting at about 2 hours post-seeding. So it's very, very early. Runs from about 2 hours to 10 hours post-seeding specifically. The one on the smooth titanium we see elongating. The cell on the macro micro titanium actually divides and then proliferates or migrates in a very random way. And the macro micro nano does something very similar. It also divides and migrates in a very random way with lots of process extension. So we took specific time points and looked at how these cells were changing shape with respect to time. And we found that the smooth titanium and the peak both promoted sort of an elongated fibroblastic cell morphology. The macro micro promoted more of a cuboidal morphology that we would use to describe perhaps an osteoblast even. And the macro micro nano, you had lots of those processes, formed a very stellate morphology. And so the 4 images down below the videos line up with the surfaces. These are all at 24 hours, kind of giving you an idea of those steady state morphologies that the cells arrived at. So it was fairly clear, despite having a similar material for 3 of the surfaces, they're all the titanium aluminum vanadate alloy, we were getting a pretty dramatic difference in cell response. So we wanted to see then if that correlated to differentiation at these early time points. And indeed it did. So we looked at 2 markers this first time around. We looked at alkaline phosphatase and we looked at ostrix. So alkaline phosphatase is an enzyme that stem cells express as they begin to differentiate to bone that's thought to help free up the soluble phosphate levels that ultimately promote mineralization with calcium in the collagen that they've deposited. Ostrix is a transcription factor, and the timing of these 2 are both fairly early. Alkaline phosphatase comes on, at the pre-osteoblast state, we could say, and then tapers off as that osteoblast becomes a mature osteoblast. Now, ostrix lags it just slightly. So our ALP data suggested that all the surfaces went up from 3 days to 10 days, with the exception of the macro micro nano. However, when we look at the mid marker, ostrix, we see that macro micro nano surface had the highest expression. So these results, even with those sort of very early 2 time points, seem to indicate that differentiation was occurring on that macro micro nano hierarchical surface at a slightly improved rate as compared to either the macro micro or the smooth. Now then, as we all know, the NIH and certainly just in the interest of good science, have encouraged us to look at more than just perhaps a single donor. So we wanted to go back and do some of this with 3 different donors. We worked with Rooster Biotechnology to source stem cells from 3 donors. We had all of them approximately age paired, plus 2 genders. We had a 21 and a 25-year-old male and a 26-year-old female. The surfaces were seeded as before, about 10,000 cells per centimeter squared on all 3 of these unique surfaces. At 7 days and 14 days, we lysed the samples and assayed a range of markers to cover both the early response through alkaline phosphatase again, the late response through osteocalcin, and a lifetime marker that is expressed by pre-osteoblasts through a terminally differentiated osteocyte, which would be osteopontin. Additionally, the whole goal with any kind of bone study is to create a mineralized surface. So we looked at alizarin red S staining to probe for mineralization of those surfaces as well. All right, so the first marker up, there's a lot of data here, so I've tried to gray out what I don't want you to pay attention to. The first marker we looked at was alkaline phosphatase. And again, we would expect that to have an early peak. And what we saw was there was an early peak at 7 days on the macro micro and macro micro nano surfaces that was significant for the nano functionalized surface, we'll say, in at least 2 of the, and all 3 of the donors, excuse me, and the micro functionalized surface in one of the donors, whereas the smooth titanium demonstrated increases between 7 and 14 days. So from a kinetic standpoint, this data suggests that nanofunctionalization was moving the mesenchymal stem cells through that early differentiation point a little bit quicker. Now then, when we look at the lifetime marker and the late marker, we see a similar story. So the lifetime marker shows increases between both time points on all of the surfaces. Most of those were significant. Again, the nanofunctionalized surface seems to have significantly more osteopontin than the others on all three donors. Osteocalcin's a late marker that's only expressed by mature osteoblasts and is that actually tapers off if you were to expand out and look at a osteocyte. And we saw significant increases again with the osteocalcin. Again, the nanofunctionalized surface seemed to be doing the best. So this is all very encouraging data. I like that, you guys can look at it and see that it's very clearly the levels change based on the donor, but all the trends are about the same. I should also mention these are all normalized to DNA. So we look at DNA and then we normalize everything back to get an idea of what each cell is producing. So it would be nanograms per nanogram of DNA. I just realized I didn't actually have the labels on the charts. And then finally, the last thing we looked at was alizarin red to see if we could differentiate between those two textured surfaces. So the hierarchical macro-micro and the macro-micro-nano surface. We looked at the alizarin red S staining and saw that the nanofunctionalized surface, again, seemed to do quite well in accumulating this mineralization or promoting the calcification response that we would like to see from mesenchymal stem cells in response to a surface. So sort of the take-home messages here, Ben, we have is that if we incorporate this macro-micro texture, it seems to have some improvement, and then adding that increased nanofunctionalization further improves things. The macro-micro certainly promoted differentiation. We also saw before that it altered migration. I didn't show you guys proliferation data, but they all basically proliferated at the same rate. So the DNA levels on all the surfaces were roughly the same. And then furthermore, when we increased the nano texture, the rate of differentiation increased as well. You also saw before that it changed the migratory and morphology behaviors of the cells. Together, I thought it was really pretty exciting and pretty encouraging. We had a fairly robust data set. The first time around, we were able to redo the entire study with 3 new stem cell lines. So we've essentially demonstrated the same thing now on 4 different stem cells, which I think is very encouraging for the field and just generating a very robust data set there, I would say. The image you see on the right is one of these mesenchymal stem cells. It's again been stained. Red is the actin, the blue is the nuclei, green is a focal adhesion protein vinculin. That's on one of the nano textured surfaces. And the grayscale part is reflected DIC and the fluorescence, and then we're able to actually sort of compile that into a really cool looking 3D image that rotates if you were curious. Finally, again, I want to thank Anna and Karina, the two undergrads that did a stellar job helping me out on this work. Also want to thank the company that sponsored the work. That was Titan Spine. Titan Spine's been acquired now by Medtronic. So we'll thank both Titan for funding the work and I guess Medtronic for funding Titan for the future. Nevertheless, we continue our efforts on these sort of rough and textured titanium surfaces and have a lot more exciting research coming along in the future. So that, I think, is what wraps up my talk for today, I suppose.
Great. Thank you so much, Dr. Brow. We now have 3 to 4 minutes for questions. You can either type them in your chat box or ask them aloud.
I can go ahead. I had a question. I was just wondering if you guys have looked at if you can get different populations. So can you start to get osteoblasts and osteoclasts versus just global changes?
So osteoblasts and osteoclasts, so the kind of the mesenchymal stem cells and the hematopoietic stem cell populations. Yes, we have done that, and I think the data is very interesting. However, that's all I can tell you at the moment. As far as we haven't done it on any of, most of our work is on nanofibers and polymers, and I haven't taken the time to work with it on the nanofibers yet. But we have done it on the metals, and the results were interesting. But that's all I can say. Let's just say, as a scientist, I found they were very encouraging, but it was viewed as maybe a little bit of a hard sell to a surgeon.
There wasn't a question. Very interesting data. Any hypothesis on the mechanism? And I think what they mean, if I can expand, is there anything about the macro, micro, macro, micro, nano that can attribute to this differentiation? Any properties about these that?
Yeah, so what I'll say is, in my view, the big benefit, and this really comes back to a lot of the other work that we do in kind of the mechanobiology space, on those nano-functionalized surfaces, and to an extent on the macro-micro functionalized surface, you're really changing the sort of mechanical state of the cell. So on a flat surface, cells can grow and form a monolayer. And when they form a monolayer, you have a lot of cell-cell connections. And those cell-cell connections tend to repress bone differentiation. In contrast, on the macro, micro, nano surface, you end up with focal adhesions. You end up with a lot of surface changes. So cells can no longer form a monolayer where they're very tight, close together. And these focal adhesions can't form universally everywhere. So you somewhat limit where a cell can attach. What that leads to is a stable focal adhesion, which causes the associated cytoskeletal proteins to also be sort of stable or mature. This leads to an increase in Rho-A activation, and Rho-ROCK signaling promotes bone differentiation. So we definitely have some hypotheses as to what's going on. Haven't probed specifically Rho-ROCK on the metals. We've done a lot of that on nanofibers that tend to demonstrate a very similar response by the cells. And we see that Rho-ROCK signaling and that mechanical linkage both play a role in improving the rate of osteogenic differentiation.
Yeah, that's pretty interesting, because very interesting to think about what those mechanisms are, and then, even thinking about how does that relate to then different donors, like why, because then if there's a connection of different signaling, then what is it about different cells that might make them respond differently? And I know maybe some of this you're already investigating, you can't answer, but I'm kind of curious, maybe if you could, I'm going to not answer my question, so I can have, there's another question, and then we can go to Higuita. We have 2 minutes left. So is it potentially possible to transfer macro, micro, nano property to other flexible material other than metal?
Yeah, absolutely. So absolutely. I think the role of hierarchical structures in regenerative engineering is of immense importance. As a PhD student way back in like the Stone Age, though I see that since Cato's giving a keynote, he might have some of my work in there. He usually does. It may have fallen out of favor with him after so long. But I worked on making these kind of hierarchical micro nano structures out of polymers. So it was a polyphosphazine microsphere incorporating a polyl lactide nanofiber structure. And we saw a lot of the same benefits. The nanostructure certainly seems to encourage cells to differentiate. The macro structure on these metals is mostly for osseointegration, so to create kind of a puzzle piece with the bone tissue, so you get a nice bind. So that part's a little bit more clinical. The microtexture and the nanotexture, though, the microtexture, like I said, keeps the cells from forming a tight monolayer with lots of cell-cell junctions, and the nanotexture sort of alters those focal adhesion dynamics. And you can certainly do very, very similar things on plastics. That's primary focus of our lab, right? So we look at plastics that are hierarchically textured with micro scale, nanoscale features.
Okay, thank you. Dr. Higuita Castro, I know you had a question, but since you're talking next, I want to give you time for your presentation. So our next speaker, as I mentioned, is Dr. Natalia Castro, and she is actually talking about nano-engineered EVs for delivery of reprogramming cargo to neuronal tissue. Thank you, Natalia.
Okay. Thank you so much for the introduction. Can you hear me? Okay, just checking. So I'm Natalia Castro. I'm an assistant professor in the Department of Biomedical Engineering, and I have a joint appointment, a courtesy appointment with the Department of Surgery at the Ohio State University. So first, a really brief introduction. In the last 2 decades, gene and oligonucleotide therapies have emerged as a promising strategy to treat a variety of different conditions and to deliver molecular cargo to the brain and potentially the CNS. There are a lot of limitations with status quo delivery methods, such as lipoplex and viral vectors. And although the research that has been conducted with these delivery methods, it has been fundamental to move clinical applications into the bedside, there are still significant limitations, such as the reliance on viral vectors, where you have a capsid restriction that limits the type of cargo that you can incorporate or deliver. You can have induced toxicity, which limits the ability to redose the patients depending on the condition. And at the same level, you have immune and inflammatory responses. And for some of these synthetic particles, you may have low permeability to the BBB. So in my lab, what we do is we use engineered extracellular vesicles. So the extracellular vesicles are, they are carriers, basically. I see them as the micro nanoletters of the cell. So they dispatch these envelopes full of molecular cargo to communicate with other cells and tissues. So what we do is we develop we develop tools to tailor the transfection and being able to actually engineer what's inside the extracellular vesicles. So the advantages of using this type of delivery method to target the CNS is that they are naturally derived. They are normally synthesized under immune surveillance because they are produced by your cells, and they have enhanced stability in biofluids because they are meant to be in circulation. And then they have an innate ability to permeate the blood-brain barrier and deliver cargo to target cells. But one of the most important aspects is that we don't have a capsid restriction, and you won't have an issue with redosing if you go with this type of therapy. So this is just a cartoon to show that when you incorporate some exogenous material into the cell is gonna pack and release these vesicles. So by fine-tuning your transfection, you should be able to design your extracellular vesicles. This is an example of a cell capturing green label extracellular vesicles from the microenvironment and incorporating the material. Then this is just an extract of some engineering EVs that were delivered to the brain, and a transmission electron micrograph of these EVs that are actually loaded with neurogenic cargo. And we're going to talk a little bit about the fabrication of these neurogenic EVs. So first, this is a work in collaboration with Dr. Gallego Perez from BME and surgery. So what we saw is that you can produce these extracellular vesicles both in vitro or in vivo. If you transfect the animal with a combination or a cocktail that has proneurogenic transcription factors, which are ASCL1, BRN2, and MYT1L. We're able to collect extracellular vesicles that have significant copy number inside of them, almost similar to the same magnitude that you would have in the cells right after transfection. And then we use these vesicles to reprogram skin fibroblasts into induced neurons. So we're seeing that after 7 days of treating primary mouse embryonic fibroblasts, we have a positive expression of the neuron marker TUJ1. So with this, we went ahead and tried this in an animal model of a stroke, and we're seeing that when we introduce these vesicles into the animals after stroke, we have an improvement in recovery percentage, reduction of the stroke volume. And then we saw that in part these this improved response can be explained by the recruitment of stem cells from the subventricular zone of the brain that is actually triggered by the vesicles that are injected. So what we wanted to do is to actually check and study the dynamics of the production of these neurogenic EVs. And what we saw, and these are studies in vitro, is that the release of these vesicles is gonna peak after 24 hours. And in these graphs, what we have is control EVs are gonna be just cells that are transfected with a sham vector. And the naive EVs are what's normally released by the cells. So we're seeing that post-release, we have a peak at 24 hours. And then something that's really interesting is we're seeing that these engineered EVs have a narrower size distribution that's actually decreasing over time, and we're studying that more closely. So with respect to the molecular cargo, we're seeing that we have an increase of around 2 orders of magnitude higher as compared to what you have in the cells after the original transfection. This means that the cell is taking this material and is actually incorporating and expressing more cargo that's packed into the extracellular vesicles. And then what we're seeing is that it correlates with the significant release at 24 hours. You're gonna have a higher number of gene copies back inside the extracellular vesicles at that 24-hour point mark. You still have a significant number all the way up to 72 hours, but the highest expression is 24 hours. Then we saw that you can actually use them to successfully deliver reprogramming cargo into other cells. And we saw that the peak on uptake for other cells is going to be at 48 hours. And then what we did is we used neuroblastoma cells to see if we will get the same effects in this type of neuronal cell. And then we saw that there is a preferential uptake of these neurogenic EVs by the neuronal cells that actually peaks after 48 hours. And then we check if our EVs were actually cytocompatible, and we didn't see any significant difference in the response of these cells. So with this, we went ahead and tested if we were able to induce successful reprogramming towards a neurogenic phenotype with these extracellular vesicles, and the answer is yes. We saw a significant change after 7 days. And we have characterization at 14 days as well, where you have a positive expression of the neuronal marker TOJ1 for this specific experiment. So this is converting primary mouse embryonic fibroblasts into this neurogenic, with this neurogenic phenotype. And then finally, we also use cultures of primary neurons to test the effect of these vesicles, because the idea is to actually have them go to the brain to treat different conditions. So we wanted to see if they were actually not cytotoxic when you were using primary neurons, and we didn't see any significant difference. They are cytocompatible also when we tested them with these primary neurons. And more interesting than that, we did some microelectrode measurements where we found that the IP activity of the cells that are treated with these neurogenic EVs is actually significantly higher than the cells that were just treated with controls. And that's something really interesting that we're expanding on right now. And then with that, I'll just move to the second part of the talk, where we use actually cell therapies. And I'm gonna show right at the end how it actually links with these engineered EVs. So to give you a little bit of context, what we're trying to do with this part of the project is to have a treatment for patients with ischemic stroke. So in ischemic stroke, you have interruption of the blood flow to specific areas of the brain, and that causes cellular deficiencies. For these patients, they can have paralysis, visual speech impairments, unilateral silent leg, and in some cases, death. One of the major issues for these patients is that there are only 2 FDA-approved drugs, but the time window to use them is around 4 hours. So if you are beyond that point, there are no therapies that will help you restore the function and heal the tissue that's affected. So what we propose to use is to implement vasculogenic reprogramming to actually provide way to heal and repair the tissue after stroke. So in this part of the project, what we do is we induce direct somatic reprogramming of skin fibroblasts into induced endothelium, and then we inject them into the stroke brain, and then we're seeing a significant improvement in the function. So this is basically an overview of the project. We use nanochannel-based technologies to incorporate the molecular cargo into the cells. And in this case, we're using a cocktail that we call EFF. It's a combination of transcription factors that are gonna guide the cells toward these pro-vasculogenic phenotype. So it's a combination of 3 key transcription factors. This is ETV2, FLI1, and FOXY2. And you can see that you have significant expression after the transfection with the nanopore-based transfection. And something that was really interesting too is that you also have a regulation of other vasculogenic factors that were not originally transfected. So after a week, we're seeing a positive expression of vasculogenic factor VWF, and we correlate that with a DDK tag that we had in our plasmid DNA, just to prove that the conversion is actually induced by the reprogramming cargo that we introduce. Then what we did is we introduced these cells into the brain of healthy animals to see if we could actually affect or improve the perfusion of the animals. This is a laser speckle image of the perfusion of the brain. And we're seeing that after a week, there is a significant increase in perfusion for healthy animals. So what we wanted to do after this, and this work was led by Luke, what we wanted to do was to introduce these cells in a stroke animal, right? So after a stroke, we injected this transfected cell into the intracranial, into the animals. And what we see is we see a significant improvement, not only in perfusion, but also in the expression of endothelial markers such as CD31, all the way up to 21 days. Something that was really interesting with this work is that you see there is a correlation between the number of injected cells when the cells are actually transfected, meaning that they are induced towards this vasculogenic phenotype, and we don't see that correlation with the control cells. So the more cells you put, the better the outcome, basically. That's the conclusion. And then for this part, something that was also really interesting was to see that the effect, a positive effect of treatment was going actually beyond the point of injection. So we're seeing a significant improvement also in the contralateral side of the brain. So that got us thinking. And then we did a study where we kind of tracked the cells that we were injecting for 6 hours. And we've seen that they actually go into the other hemisphere, but it wasn't as significant. So we did a study where we actually isolated vesicles of these cells after they were transfected. And we're seeing that the vesicles that they produce, which we're gonna call the engineered extracellular vesicles, are actually also packing these key vasculogenic markers that we were seeing when you transfect directly the cells. So what we hypothesize is that these extracellular vesicles or engineered vesicles are actually going beyond the point of injection and improving the outcome of the animals, even to the other hemisphere. And this is just to show the recovery. We have a significant improvement in the reduction in the infarcted volume. After 21 days, this is 14 days after treatment, 21 days after stroke. And then we also see that there is a significant amount of neuronal content and less astroglia formation, as you can see with the micrograph.
Have 1 minute left actually in your whole time. I forgot to stop you.
Okay, so with that, I'll just show you these videos real quick. We saw an improvement in the locomotive ability of the animals. So you can see that the control animal, it's only has a really restricted range of movement. And the animal that's treated with the vasculogenic cells and EVs is freely moving around the field of view. That I will move to my last slide. We have, these are all the parameters that are actually improving with the therapy. They go faster, they travel longer distance, they have more time mobile, And they have a lot of significant improvement in counterclockwise rotations, which is really significant, for example, in a clinical setting, because it correlates with the side neglect that the patients can have. So with that, I will just conclude. For the first part, we saw that we're able to successfully characterize the fabrication of these pro-neurogenic EVs. We're seeing that the production peaks at 24 hours, the uptake peaks at 48 hours. So we're seeing the potential that the designer or engineered extracellular vesicles have to actually effectively transfer this molecular cargo into the cells and induce reprogramming. And then in the last part, we saw that we can introduce these reprogrammed cells that are reprogrammed towards a vasculogenic phenotype into the stroke brain, then improve both perfusion, infar volume, neuronal content, and functional outcomes for animals after stroke. And we're seeing that the engineers that are produced after transfecting those cells with the vasculogenic cocktail are key to diffuse or transfer those repair molecules into the surrounding tissue. So with that, I'd like to acknowledge our collaborators, Dr. Gallego Perez, my lab members, and the funding sources. And thank you so much. Thank you for your time.
Yeah, thank you.
Stop share.
I think we may hold questions for later just to keep on track. I apologize for not giving you a little bit of an earlier warning, but I think please ask if you have questions, put them in the Q&A. And I think Dr. Natalia Castro can stay afterwards to answer those questions. I'd like to introduce our next speaker, Dr. Jennifer Pitzer, and she's going to talk about driving hierarchical collagen fiber regeneration for functional musculoskeletal engineering. Thank you, Jenny.
All right. Thank you very much. And thank you for inviting me. I'm very excited to talk to you guys today about our work on producing strong collagen fibers to try and repair tendon, ligament, and meniscus. So why do we care about collagen fibers? Collagen fibers are our primary source of strength in tissues throughout the body. And this is particularly true for our musculoskeletal tissues, such as the meniscus, tendon, and ligament. So for those of you not familiar with the meniscus, it's basically this semi-lunar wedge-shaped disc in your knee that makes it so it's not painful when you walk. And of course, our tendons and ligaments connect our muscles and bones throughout the body. And what these three tissues all have in common is that they're composed of these large type 1 collagen fibers that run the length of the tissue. So in the meniscus, they run circumferentially. You can kind of see that here in this blowout. While in the tendon and ligament, we have that traditional hierarchical structure that increases in size until it's basically the entire tissue. And so these collagen fibers are what provide all the strength and function to these tissues. And so whenever we tear or injure our tissues, it disrupts that collagen organization, makes it so they're no longer functional. And unfortunately, cells just really can't regenerate these fibers again. And so this often results in surgery. And so us as engineers are really interested in trying to understand how can we create replacements for these tissues. However, we're often unable to create enough collagen, let alone the organized collagen that's essential to their mechanical success. And that organization that we're really after is the hierarchical structure. So basically, we start out with the tropocollagen molecules that are what the cells create and spit out into the matrix. And once that's out in the matrix, the cells will start to organize it into microfibrils. Those microfibrils get organized into fibrils. Fibrils are typically nanometers in diameter. They then will group those fibrils together to form fibers that are often in the micron range. And then finally, in our musculoskeletal tissues, we see these fibers grouped together to form much larger fascicles that can be hundreds of microns to millimeters in diameter. And so currently, the field is really well versed on how we can regenerate this fibril level organization, but it remains much of a black box of how do we move to these larger fibers and fascicles. And that's really where my lab sits in trying to understand how do cells do this and how can we reproducibly get them to make those over and over again. So the way the field has gone after trying to create fibrils is primarily by harnessing cellular contraction in low density collagen or fibrin gels. So basically what you do is you seed a whole bunch of cells into a collagen or fibrin gel, you anchor it on either side with either posts or sutures. And then since cells are always in contraction, as they start to contract, they feel those posts and they end up aligning the fibrils across the tissue. And so this works really well for getting nice aligned collagen fibrils, but because these are such low density, they're quite weak. And so we have very significant contraction. You can see down here to the suture level. And the cells just aren't able to mature these fibrils much further. So during my PhD in a whole meniscus model, and then during my postdoc and now in my lab, in a more simplified model, we've been able to demonstrate that if you just shift to a higher concentration of collagen, so 10 to 20 mg per mil instead of 1 to 5, we can actually support and stimulate these cells to get them to move past the fibril level and start to form larger fibers and fascicles. And so today I'm going to talk to you primarily about our work in developing this system and starting to push it further. So when we first set out to develop this system, we were basically interested in developing a simplified, more thorough system so that we could really look at driving that hierarchical collagen fiber formation across multiple tissues. And so what we do is, of course, we extract and reconstitute all of our collagen from rat tail tendons. And for everything I'm showing you today, we're working at 20 mg per mil. We then mix our cells directly into our collagen while it's still liquid, so they're spread throughout. And what I'm going to talk to you today about is work where we were using primary bovine-derived cells, where we either got tenocytes from a flexor tendon, ligament fibroblasts from the ACL, or fibrochondrocytes from the meniscus. We then cast that sheet gel, allow it to set up, and basically take a little cookie cutter where we can cut out rectangles and then culture them in our device. And so this fits into a 6-well culture dish. We can screw down, clamp our tissues in place, and these clamps then will help to stimulate the cells to align the fibers across the tissue. So I always like to show this picture because it kind of shows our cells in action, right? So no matter what, if we put tendon, ligament, or meniscus cells into our gel and it's unclamped, we get this significant contraction with time and culture, and by 6 weeks, it's basically nothing. While if we clamp our tissues, you can see we maintain that size and shape, and we're really harnessing that contraction power of the cells to drive our fiber formation. We do a lot to look at our overall fiber formation. And so here we're just using confocal reflectance, where the gray is collagen and the green is cells. And you can see in our unclamped samples, we really have no organization. It's just a real compaction of that collagen. While if we clamp our samples, whether we put tendon, ligament, or meniscus cells in it, by 2 weeks, we get this nice alignment of fibrils throughout. They're so small that it's hard to see, but it's basically those streaks. And then with further time and culture, we see these really grow in size. And by 6 weeks, we start to see distinct fibers that actually match their native tissues. So in the tendon, we see much smaller fibers, more wavy, while in the ligament and meniscus, we're seeing larger. We, of course, wanted to prove these aren't one-hit wonder pictures. And so we've done a lot of image analysis of this. And so in these graphs, our solid bars are our clamped tissues, with black being tendon, green being ligament, and blue being meniscus. And then these dotted lines are their native controls. And so we found that we're able to match native alignment by about 2 weeks. And then when we look at mean fiber diameter of our tissues, you can see by 6 weeks, we do in fact get significant differences in those fibers. So our tendons are making the smallest at about 20 microns, ligament the next largest at about 30, and meniscus the largest. And so this is really interesting to us because here we have these cells that often people might just refer to as fibroblasts. We put them in the same environment and somehow they intrinsically know to make their base fiber sizes. So we're currently starting to look into exactly how do the cells know to do that, so that hopefully we can either push this in vivo after injury and get them to actually regenerate these fibers, or just be able to actually make these tissues reliably as replacements. We can then zoom out and look at the next hierarchical level. So here, these smaller fibers that you see in the meniscus are what we were looking at before. And now we have these much larger fascicle formations. And you can see the same trend holds up, where the tendon at 6 weeks has the smallest fibers at about 50 microns, the ligament the next largest, and the meniscus at the largest at hundreds, about 300 micron wide collagen bundles. And of course, paramount to all of this organization is the overall mechanics. And so you can see here that with that organization, we see a significant improvement in our tensile modulus to about 1 megapascal by 6 weeks. And this is in stark contrast to if we look at our collagen that we have in our constructs. So we do this by hydroxyproline, and you can see that that really just doesn't change with time and culture in our clamp samples. And this just really demonstrates the need for that underlying organization to get those mechanics. So overall with this study, what we have found over and over again with many different cell types is that using this culture system, we can drive native-like hierarchical structure with it at the fibril, fiber, and fascicle level. I didn't show it, but we have zoomed in and looked at the fibril level to see if we were getting that native banding pattern of the collagen back, and we were. So we're pretty excited about these, but these are some of the largest and most organized fibers produced to date in vitro, but there's still a lot of work for us to do, right? We can kind of already see that the fibril level isn't as good as we would like it. And then of course, when we look at the fascicle level, if we were to zoom out and look at the next level up, we'd see there's a whole nother level of fascicles we're just not getting. So currently in my lab, we're doing a lot of work to look at how can we make these stronger and how can we make them bigger. So one way that we're looking at trying to make them stronger is by looking at the natural cross-linking of collagen. So collagen is naturally cross-linked by the enzyme lysyl oxidase, or LOX, and basically LOX will go in, form an immature divalent cross-link, and then with time, for unknown reasons, in our really strong tissues, this will condense to a mature trivalent cross-link. So we're really interested in understanding what actually drives that change, and can we use that to either make really strong repair tissues or to make our engineered replacements. And so our student Madison has been working on this, and one of our first thoughts was maybe it has to do with the underlying organization. So she's been culturing our tissues out and basically dosing them with LOX at different levels of organization. So we dose it with LOX at 2 weeks when we have fibril level organization, 4 weeks when we have fiber level organization, or 6 weeks when we have fascicle level organization. And what we found so far is that if we dose those at the fibril or fiber level, you can see by 6 weeks, we get this significant improvement in our mechanics, getting up to about 30 megapascals by 6 weeks, which is getting us into that immature range of tissue and making these a bit more functional to go in as a repair. We're then also interested in how can we make these fibers bigger and stronger at the same time. And so we're doing that using mechanical stimulation. This comes from the idea that, of course, when you're born, you have more fibers kind of like this where they're smaller. And then as you continuously load your tissues throughout your adolescence, your fibers grow and get bigger and stronger. So we know mechanics is really important to driving this process, but there's a lot of mechanical signatures. So we're interested in figuring out exactly which mechanical loads are the most important. So currently what we're doing is we're taking those samples, putting them into our bioreactor, and then basically dissecting out those loads. So we're doing a slow stretch that mirrors kind of your growth and the elongation of your arms and legs. We're doing quick cyclic loads that mirror you quickly moving around your arms and kicking your legs as a baby. And then, of course, combining those together to see, can we push these to adult size fibers? And so we're just kind of diving into this work and haven't done too much on the main fibers, but we have had some great success with this in our interface work. So basically, wherever you have a tendon, ligament, or meniscus in the body, it has to anchor into bone. And this anchor point is really, really important because you have to be able to take these loads and transfer them from the soft elastic tissue to the stiff bone, right? And so that mismatch can lead to a lot of injuries. And the reason our attachments actually can handle this and transfer the load is actually because they're super highly organized. So wherever we go into bone, in about a millimeter, we go through 4 different zones. We go from highly aligned ligament fibers to smaller collagen fibers, less organized fiber cartilage, to kind of mineralized cartilage, to finally bone. And these zones are what are so important to really transferring that load. So we were really interested in seeing, can we regenerate these zones so that we have functional replacements that can anchor to bone actually. And so in our lab, a lot of what we like to do is look at how does it develop naturally and can we repeat that? So we looked at how does this develop naturally? And it turns out that basically when you're first born, you actually don't have any of these zones. It's just unorganized tendon or ligament going into cartilage. Then as you start to load these a bit more, you go through this phase where you have a line ligament, small fibrils in the ligament, completely perpendicular fibers to that in the interface, and then cartilage turning over into bone in the bone side. So we looked at this and we thought that our constructs, our cultured device could actually help us get this, right? Because basically in this midsection, we already know we go through this process thanks to the tensile loads from our cells. And then it's pretty well established in the tissue engineering world that if you apply a compressive load to your tissue, you can drive cartilage formation. So we thought that possibly this clamp could start to help to drive that cartilage side of things. And so I'm going to condense down my PhD's work for the last 3 years to a few pictures. But basically, we've been looking at this and we find, of course, at day 0, we have no real organization in our mid, transition, or clamped regions. If we clamp these for 2 weeks, we start to get that early postnatal-like organization where we have aligned fibrils in the midsection, completely perpendicular fibrils in the transition, and more cartilage-like tissue in the clamp. And of course, Ethan, my PhD student, has done all this great work to show that we also are getting different biochemical accumulation that matches that, where we have GAGs in the right spot, collagen 2 and collagen 10. So we really think we're starting to get this early postnatal shift. And then of course, we want to keep improving this and moving it towards that native level. So we've added a slow stretch to this for 10 days that mirrors that growth rate of your ACL actually after you're born. And what we find is we get a bit better fiber formation in the mid region. In our transition region, these fibers are actually shifting to go back the other way as we would expect. And then a bit more bone-like formation under the clamp. So we're really excited about this progress so far, and we're currently starting to look at if we add cyclic load to this, what does it do? And then of course, combining it together to get to the hopefully adult level strength. And we hope that this will also inform not only engineered tissues, but in ACL graft repairs, trying to regenerate that interface anyways. So basically, just to wrap up, this is kind of the different areas that my lab works in. Of course, we're really interested in driving fiber formation and making them bigger and stronger, trying to integrate these into bone. We're doing some work looking at beta tricalcium phosphate stimulus to help with that bone formation as well, with our mechanical stimulation. And then we're also doing some work looking at using this as a way to look at how do things change with aging, and hopefully be able to mitigate those changes to reduce injury. And our overall goals are, of course, to engineer replacements that hopefully can go in one day, but at the same time, learn how our cells are changing and hopefully be able to stimulate that to drive regeneration in vivo. And so with that, I will, of course, acknowledge my students and our funding sources, and I'm happy to take any questions.
Thank you. That was a great talk, and we now have a few minutes left for questions. Enter them in the chat box, Q&A, or say them out loud.
I also think I started a bit early, so
you did.
Questions for Natalie, you can go to.
I was talking with Natalie after this, so I was misreading the time. That was when her talk ended, so she was perfect, and there was a built-in time I just did not recognize.
That's fine. I thought I was reading my timer wrong, so it was like, okay, yeah.
No, you were fine. It was all me. So
Okay.
I apologize. She's agreed to stay after to answer her questions.
Yeah.
Thank you for recognizing that.
So Jenny, this is a very nice talk. Maybe I'll start with a question. So fascinating work. So One question is, in your experimental model, you put cells into existing collagen, then ask the cell to align them. And then in the native tissue, I think when the collagen getting secreted by cells, they're being aligned, right? Now, how would you compare this? Seems like in in vitro, it's amazing you can achieve this because there's a lot more work for the cell to do, right?
Yes, yeah, definitely. So yeah, that is a really great point. A lot of times, basically, when we're developing, we first form those attachments and then the cells sense that and lay down the collagen. So we are asking them to do a bit more work and that they have to go unorganize that mess and realign it. But it seems like they're okay with it so far. We do have to vary those concentrations depending on the cell type. So with our tendon and ligament and meniscus, they're strong enough that they can go do that, and that's not a problem in the long run from what we're seeing. But if we were to we've done some work with periodontal ligament and gingiva, a little less sensitive cells, and this level of collagen is just overwhelming for them to sort out. So in fact, with those, if we go down to about 6 mg per mil, we can get them to drive their natural fiber formation. So I think it's just all a balancing act to what these cells are able to put up with and organize.
Thank you.
So I have a question. It's a really interesting clamp system that you have that you seem to, really well characterized, and it really works. I noticed there's like a little bar kind of in the middle. Does that height matter at all? Like, what are the, more broadly to the properties of that clamp? It looks simple, but I'm sure it's really well engineered.
Yeah, so we basically put about 1 millimeter step down here to our clamps. And that is where we have a bit of a difference from those low density systems. And we do this just to get some more mechanical strength when we're doing this, because those cells are quite strong as they start to align and start to contract. And in order for these tissues to actually hold on and not slip, we need that kind of step down to give it a little more mechanical load. And if it was just flat, they would just pull free. We also have meshes in there to help with not allowing them to slip. And so these are all things that help support these cells to move past that fibril level to those larger fibers of fascicles.
Yeah, it sounds like something that, again, you're presenting it as simple, but probably took a very long time to figure out. And then there was also a slide you had before where it looked like they were kind of contracting in the middle. What could you maybe explain a little bit what you think is happening there? Is that like physiologically relevant, or just?
This kind of relates to Charlie's question, but we've done a lot of work to kind of look at what are the cells rearranging the collagen, or basically getting rid of the collagen and then laying down their new collagen. And this is a really hard question for us to answer because collagen is preserved across species. So you can't really separate out the rat tail collagen you started with and the cow collagen that the cells are making. So we usually look at it by looking at what's in the media and how are our constructs changing and all of that. And from taking that all together, what we know is that basically in those first 2 weeks when there's not a lot of contraction, the cells are basically just spitting out tons of collagen into the media. So we think during that time, they're basically just kind of throwing out the collagen that's around and laying down their new collagen. Of course, that's a hypothesis. We still haven't been able to completely prove that. But then once they start to contract in, that's when they're starting to kind of just pull collagen from around them to build up those fibers a bit more. So we expect to see that happen. It's not the end of the world. It actually gets us closer to ACL size. So it's somewhat handy.
Okay, and last question before Dr. Ren, any idea about how to implement this into patients, like how to reproduce the clamping effect? I'm assuming clapping is clamping.
Yeah, no, that is a great question. So basically, if we right now, the way that you would repair the ACL, if we stick more with bone, it's easier. I mean, ligaments than tendons, it's a little easier. But the way they do that is basically they will drill holes on each side in your bone and then take a graft of a tendon and put it through that and clamp it into the bone. So we could pretty easily do basically the same thing and clamp our ends of our tissues into the bone. For the most part, I know bone people usually don't like you to say that bone can regenerate, but in comparison to tendon, ligament, meniscus, bone we can rely on to regenerate pretty well. So if we're not too concerned that in those tunnels we can get bone, and the main thing is getting that in between and those connections that we're concerned about. And so we think those anchors into the bone are what will help with this.
Very exciting. And as someone who went to grad school with you, good to see how well you're progressing as a professor. Really exciting. Our next speaker is Dr. Charlie Wren. Dr. Wren will be talking about recapitulate human cardiopulmonary co-development using simultaneous multi-lineage differentiation of pluripotent stem cells. Let's all welcome Dr. Wren.
I'm mute myself. Thank you, Liz. And thank you for the organizer for inviting me here. So it's really a pleasure to actually, for the first time, to talk about our recent work on modeling cardiopulmonary co-development by using human stem cells. So before I start, I really want to shout out to Waiho, who is the driving force in our lab in the past 3 years on this project. So now I'll get started. So before we talk about our work, I just want to show everyone, probably, most of you are aware, the heart and lung are very closely connected. In our chest cavity, they are the two main organs, and actually they are very closely connected ever since the embryonic development. So if I were to show this short video, this is a mouse embryo. It's undergoing development on day 12, day 13. And if you start look at this circle, you see a little triangle-shaped structure going out. This is the lung. So basically, if we look at this in zoom in, so this big chunk is the heart, and right next to the heart is lung. So they're very, very close together. So of course, the heart is one of the most important organs, show up the first. Then lung immediately follow the heart and right next to the heart. And also we're summarizing some of the work that people have done in the mouse model. We find that not only they're close to each other, there is actually a lot of signaler communications between the heart and lung. For example, people have shown that the heart field cells can secrete a Wnt ligand to act on the lung when the lung is still, before it become a lung, is a foregut endoderm, actually to drive the lung differentiation. And then when the lung differentiates, it's going to secrete sonic hedgehog, another kind of paracrine signaling, going to come back and act on the heart field cells to promote atrial septation. Actually, when this signal get messed up, the heart are actually going to have a septal defect. So because of this, we're thinking about, how can we make a stem cell model that we can study this in a defined system and potentially human specific. When we look at the current models, right now, because of this has been a lot of progress on stem cell differentiation, now we can generate heart cells or lung cells with very high efficiency from human pluripotent stem cells. However, so far there has been minimum model that you can study the between organ interactions using stem cell model. So if right now you will want to study a cardiopulmonary differentiation where there are interactions, then you would have to get help from our little animal friends. So what we're trying to do here is, can we potentially come out with a stem cell based model that we can drive two lineages together at the same time so that we can study their interactions? So, and then potential benefit, number one, you're doing a Petri dish, it's a more defined system. You can manipulate signal cascade much easier. And then it's humanized because you can study human cells, human biology, and that make it potentially more translatable to future regenerative medicine if we find out that the model can help us to make a better lung or better heart. And then, frankly speaking, sounds a crazy idea when I just started in my lab 3 years ago, and when our lab started actually 3 years ago. So because we all know that the heart and lung coming from different germ layers. So we know germ layer specification is one of the first things happen in development. We have 3 germ layers, people believe, ectoderm, endoderm, mesoderm. So the heart is coming from the mesoderm, the lung is coming from the endoderm. They literally say goodbye to each other at the very beginning. But we do catch some interesting phenomena. We realized even though they are coming from different germ layers, but they share a lot of requirements of the same set of signalings. For example, at the beginning of the specification, they all need Wnt, they all hate insulin. And then in the middle, they all need to inhibit Wnt again. And then later, they all need retinoic acid. So this motivated us to say, well, if we can get initially specification of endoderm and the mesoderm, there is actually a chance for us to get both heart and lung at the same time. So, and then of course, the beginning is we have to get a mesoderm endoderm. That's something we cannot go across because heart is from the mesoderm, lungs from endoderm. So we're fortunate that we were able to just manipulate one single signal of the Wnt using small molecule Wnt activator CHR 99021 at the middle concentration. As you see, we got really robust induction of the endodermal marker together with the mesodermal marker. However, if you go lower concentration, you get little induction of both. When you go too high on the Wnt activation, you get no endoderm, but a lot of mesoderm. Again, all those phenomena matches really well with what people have learned from mouse embryonic development, that because it's a gradual transition, too high of Wnt will favor mesoderm, but both endoderm and mesoderm require a certain amount of Wnt. And then encouraged by this, so we came out with a stepwise differentiation model. It takes about 15 days from IPS, hopefully to get it towards a cardiopulmonary program. So the 3-stage we set is closely mimicking the development cascade in mouse. So the first stage corresponds to germ layer specification. Second stage corresponds to anterior foregut specification and also cardiac mesoderm induction. And then stage 3 corresponds to cardiac and pulmonary induction. So actually after, I guess, while we have done hundreds of optimizations, we were actually able to come up with this protocol that we can drive really robust co-differentiation of both heart and lung. So the green labels NKX 2.1, labels the lung progenitors. The red NKX 2.5 labels the heart progenitors. And then we show that, really the induction of the heart and lung really echoes the induction of mesoderm and endoderm. If you remember, in the mesoderm and endoderm induction, we see really balanced induction of both species at the middle CHR, 7 micromolar. And same thing we see here. When we give 7 micromolar CHR, we get balanced induction of lung and heart. However, if you go too high, you get only heart, but no lung. Again, echoes only mesoderm, but no endoderm. If you go too low, you get nothing. Again, consistent with the germ layer induction, which was confirmed by those qPCR results. This really, I was trained as a developmental biologist, so that really made me happy because I think that echoes to one of the principles we learned from developmental biology is the spatial coordinate that a cell or tissue has in development determines what kind of signaling the cell will get exposed to. And because the heart and lung are so close to each other, it's not surprising actually, the same set of signaling can drive the co-differentiation of both heart and lung. So, and then the next, we need to solve a question when we start to discuss this project with other people in the lung field is, we all know that, traditionally when people try to induce heart and lung differentiation, they need to use TGF-beta signaling. But in our case, we do not introduce TGF-beta. So that's why we study how exogenous TGF-beta would do in our system. So in the middle group, we introduce additional TGF-beta in the form of activin A ligand, or we inhibit TGF-beta using a small molecule inhibitor. So we find that if we introduce more TGF-beta, the lungs stay the same, but the heart is gone. If we inhibit TGF-beta, both heart and lung are gone. So this is confirmed again by qPCR. So our theory is that you need certain baseline level of activin A level or TGF-beta level to enable heart and lung differentiation, which is provided endogenously in the differentiation system. If you add more on top of this, you would further promote lung and then abolish heart, which is again consistent with what people believe in embryonic heart and lung development, that higher level of TGF-beta signaling favors endoderm at the expense of mesoderm. And then the next is, once we have a cardiopulmonary induction program, we want to mature them to be functional tissues, right? So we adopt two models. One is a suspension culture, which is going to give us organoid. You see the lung cell green in the center, the heart cell is on the outside wrapping around them, or in something we call air liquid interface model, where the monolayer is exposed to air. So this is one of the very commonly used technique for lung stem cell research. So, and thanks to our collaborator, Derek Cotton and Finn Hawkins from BU, we got a transgenic cell line that they label the green color with the lung progenitors, label the red color with whenever the lung cells become alveolar type 2 cells. So interestingly, we see that in the 3D suspension model, we get really robust alveolar induction in 3 days, which based on literature should take 10 to 14 days with what we can find right now. However, we don't see much induction in the air liquid interface model, indicating that the suspension model is advantageous. And also interestingly, like I said, it's much faster than what people have reported when people study lung cell again without heart cells. So then we want to answer the question, does the heart actually being the reason of the magic, that we can get much much faster induction of the program. So we set out a control experiment where we make a dual lineage organoid with lung cell in the center, heart on the outside, or we can simply make a single lineage lung organoid just by boosting the activity at the beginning. Then you would remove all the heart cells. So indeed, when we have the co-differentiation model, we see this robust alveolar differentiation in 3 days. However, we see a little bit, but we don't see very much when we have a single lung lineage organoid. So really indicating when you introduce a critical cardiac niche, you can really expedite the alveolar maturation. And then the fast alveolar maturation is something what we have been intentionally look for. But here is something where that we come across that we didn't specifically look for, but just really come to us as a surprise that, like I said, when we first form the organoid, the lung cell is always in the center, the heart cell on the outside, as you can see on staining here. However, over time, I think these two tissues realize they don't belong to each other. They actually decide to leave each other over time. As you can see, this single organoid time-lapse imaging, the green is the lung cells, the non-green is the heart cells. So you see the lung cells gradually budding out, and then eventually they separate from each other. There's no phenomena we've seen before. So so far we call it cardiopulmonary tissue segregation, which is captured by those histological staining. So again, the green is heart, so the green is lung, the red is heart. So this is, again, I want to echo back on developmental biology and tissue anatomy. When we take a cross-section, or whenever a student do an anatomy course, when you open the body, we'll be amazed how crowded our body is. There's all kinds of organs crowding a very tiny space. But, we usually rarely see one organ merge with the other, because once that happens, it must be very bad to that individual. So, but I think potentially our model is providing us a way to study the mechanism we call tissue segregation with tissue boundary formation. Seems like different tissues know that they need to set a boundary between them. And then we want to study the mechanism in more detail with help from Dr. Yuli Wang and his postdoc David Lee. So we study in particular how Wnt signaling affect this process. We realize that once we activate Wnt with the C drug, which corresponds to the canonical Wnt agonist, there is segregation, but it's very slow. However, in this purple color group, when we remove the activation of Wnt, we get much faster segregation, indicating that the canonical Wnt is slowing down the segregation process. And once you remove that, you will expedite the lung leaving the heart. We also study other signalings. For example, inhibition of Wnt, either canonical or non-canonical, it does not seem to play a major role. So lastly, as we see the cardiopulmonary tissue start to segregate, we actually see a concurrent start of the cardiac maturation indicated by the cardiac contraction, for which we get help from Dr. Adam Feinberg and the PhD student Jackie Belaley to help us to characterize the cardiac phenotypes. For example, we can see after segregation, the cardiac organoids start to contract, and then they start to show a cyclic calcium signal handling, which we can measure with image analysis. So with that, that's all we have for today. So I want to thank the lab members, the current member and the past member, and Waihuan Liz especially, and then also our collaborators and our funding source. Thank you.
Thank you for that wonderful talk, Dr. Ren. Now we have some time for questions. If you have any questions for Charlie, please put them in the chat box or say them out loud.
I have a quick question, actually.
Absolutely.
Thank you so much. This is a wonderful talk. I was wondering, I don't know if I missed it. So do you think that the cells need to be in contact to have this effect? Or do you think that you have effect of some soluble factors or exosomes or EVs that are actually guiding the results that you're seeing? So if you were to put the cells in a co-culture, but in separate spaces, but then just sharing the media, do you think that you would have the same effect, or have you checked for that? I just think it.
We haven't. I think this is a great idea. Thank you. I think exosome or secretive factor definitely account for a big chunk, even majority of the mechanism. We haven't done any experiment on that yet, but I think this is a very good idea, and I will be happy to look into that. I think EV is really rising as a major communication mechanism between cells, and then an EV of different size, or even some secreted factors free from vesicles. So I think that will be very interesting to look into in the future.
Yeah, pretty neat work. Thank you.
I have a question. Very interesting work again. I was interested in, do you guys have any guesses on why you're able to do this so much quicker in suspension versus that air liquid interface?
I think the suspension culture, I think is created a 3-dimensional model. So the mechanics of the substrate is being different. When we do the air liquid interface, the cells was cultured, I think, in a type of polymer or plastic like mesh. So it's still relatively 2-dimensional, which is create a thin coating. I think if you want to get more biomimetic or more expedited tissue maturation, 3-dimensional model seems to be a more feasible technology in many cases. I think that's one of the driving reasons.
What do you think some of the, I guess, applications of this would be, these results?
Yeah, thank you, Liz. So I'm thinking very hard on that, especially when I write grants. So
I'm not a reviewer.
So I think there's a couple of things. So number one, I think we provide a model that we can study the congenital disease that's affecting both heart and lung. For example, there's a disease called a congenital diaphragmatic hernia, CDH, is because the abdominal organ enter the chest space and squeeze on a lung and heart, they can become underdevelopment. So we can potentially provide a model for that. And then more on translational side, I think one of the exciting data we see is on the lung side, is that number one, we can induce lung differentiation with the help from heart in a growth factor-free condition, you don't need any proteins. So this is, to our knowledge, the first report. The second is we can do lung maturation in suspension culture. This is also the first we're aware. So the prior models, people always do a hydrogel embedded culture, the embedded organelle in the hydrogel. Again, from translational perspective, if we want to regenerate a human lung, which I did that in my postdoc training, you need hundreds of billions of cells. So, and then the embedded culture or a 2-dimensional culture is not going to satisfy the need of the cells. So I think by demonstrating it's possible to generate functional robust lung cells in suspension culture can potentially have an impact on the cell therapy and tissue engineer space.
That's pretty powerful. That's pretty interesting, right? I'm really excited about the organoids for some other reasons as well, because we're working together.
Exactly.
I wonder if this has any kind of also implications for like pathological reasons. So you were talking about development, and I was just thinking about cancer metastasis in particular, when you're talking about the organs being so close, and the idea that some cancers like colorectal tend to metastasize. It's like a hybrid of invasion. So they invade through this space and just are able to get inside the entire cavity because they're close, and they're just kind of in that ascites fluid. And I don't know, I mean, I don't think you studied this, but it could be interesting to think about what are some of the relationships of connection in space that maybe are happening after development. In particular, thinking about cancer, which tends to defy all, have all regions of regeneration potential.
Yeah, I think
this metastasis capabilities, and in particular, I think in colorectal cancer, any of the cancers that are kind of in the abdomen space that tend to spread by fluid.
This is a great point, Liz. Actually, I never thought about this until you say this. So right now, because encouraged by this, we're trying to mix everything together. We're trying to get this, make this tissue organoid, make that tissue organoid, merge them together. What we realize is they kind of attach to each other, but they don't really merge. They don't encapsulate. That means the cell has the repulsion not to get mingled with another type of cell too much. That's, we say, healthy cells. But it will be so intriguing to see whether the same thing holds for a cancer organoid. If you were to merge a cancer organoid with another benign cell tissue, would the cancer be more invasive to very active go in there? I think that we may actually have a metastasis model. This is a great idea. And of course, then we're going to come up with ways to stop them. Yeah.
Or just like, why does it make them more metastatic, or
right.
Do they evolve in these other ways? So I want to now give space for us to go over Dr. Natalia Higuita Castro's questions. If people are still here who have questions, I think this is the general portion of our session, but I'd like to open this back up because we missed the wonderful opportunity to have questions. I will actually start so people can put things in the chat box. And I'm going to start with what I think is a basic question in the exosomal kind of field. When I talk to people about exosomes and the power of exosomes, they often think, well, like, how is it being targeted? And so even though look, the exosomes go to the tumor, the brain or any other inflammatory disease, how does that happen? So I guess if you had any kind of insights into how do you think these exosomes are targeting, or what's on their receptors that are making them be so specific to stroke?
That's an excellent question. So normally they are a snapshot of the donor cell. So they are going to have whatever molecular cues you have in the donor cell on the membrane, and whatever molecular cargo you have from the donor cell. So I think part of that is going to increase the affinity depending on the donor cell. So whatever the tissue source where the extracellular vesicles or exosomes are derived from, it could increase the affinity to a specific cell or tissue niches in the body. Through engineering, we're able to fine tune whatever it's expressed on the AV to tailor that targeting. So yeah, that's a great question. Thanks.
And were you, what was the source of your exosomes? It seemed like you were injecting something into the mouse, but then, so are the EVs coming from, I guess, just all cell sources?
They can, yeah, but for these two studies that I was just showing, they are derived from fibroblasts from the skin. So we can get them, generate them in vitro or in vivo. So the ones that I was just showing, they are fibroblasts derived babies.
Awesome.
I had a quick question. If I remember correctly, you had the size decreasing with time, right?
Yeah.
What are your ideas for what's going on? Are they producing less, or they're getting better at packing it?
I think it kind of, so the dynamic of packing of the three different factors that we have is changing over time. I think it correlates with the size of the gene. So we have genes that go in the cocktail. There are three factors. So one is 10 kb, the other one is 8, and the other one is around 7. So I think as the dynamics of packing of those three factors starts changing, there is one that significantly drops. So my hypothesis is that correlates with the decrease in size, because you have less packing of that one factor in the AVs over time. And that's just because of the expression profile is just different for the three factors. So yeah, that's a great question. Yeah, thanks.
Okay. And so
I actually have a quick question for Jenny, if I can.
Awesome.
Just
Let's
real
do it.
quick. Sorry, I was just wondering if you have thought about having your device implanted sub-Q or flank of the animals, and see if having all of the surrounding environment, it's gonna improve, or what effect it's gonna have in the organization of the fibers.
Yeah, so we haven't done that yet from the low density work. So a lot in the tendon and ligament world has been done with like 1 to 3 mg per mil. And usually with that, they see like within a few days, dramatic increases in amount of collagen and strength, right? So I'm pretty sure that if we did start to put these in, we would be able to really significantly improve that organization. So we haven't quite done that yet, just because we're doing a lot with bovine cells and, like the whole translational pathway. It's just messy. So we like to think about this more as kind of a model to understand things a bit better. And what we're really concentrating at the moment is kind of those mechanical loads. How do they do that? And then hopefully we can start to inform physical therapy so that we can say, like, after you tear your ACL, it's really good if you do these slow stretches. That's what's really going to drive it back. Or is it slow stretches with some cyclic load? So that's kind of our more immediate at the moment, because hopefully we can actually cause some change a bit quicker while we still keep an eye on developing these up.
Awesome. Thanks.
It's ambitious, but I think we all should make a big grant, where we use those macro, micro, nano structures on your clamp device that, you clamp into the bone, put some exosomes in there, put it next to the heart. And then we'll just
The
make
ultimate.
it. But in the meantime,
DOD, full regeneration.
Yeah.
in the meantime, we will all just try to get funded and stay sane during COVID and keep everyone happy. The next session is the NEBEC Regenerative Engineering Keynote by Dr. Cato Lawrenson from the University of Connecticut. That will be held at 11:30, which is 5 minutes from now. We hope you will all join us there. So this is the formal end of our session. If you have other questions, maybe the speakers will hang on for a second, but otherwise, we hope to see you at the keynote lecture. Thank you for coming to this session.